In this lecture, we're going to talk about Chapter 17, and Chapter 17 is a process of translation, and there's a lot of detail here in the textbook, and the goal of this lecture is to just provide just a minimal framework of what is going on. And so we're at the ribosome in the cytoplasm of any cell, and the ribosome is bound to an mRNA and is copying that into a protein. So that's what's going to be happening, and the the information is coming from the transcription unit, and the transcription unit for proteins that code for genes that code proteins is got this coding region. The coding region is the genetic information that tells the specific structure of the primary amino acid sequence of the protein, like that. Okay. So we've been talking about that, and here it is in Chapter 17 showing how the DNA is double-stranded and it is going to get unzipped, separated in order to get copied. So one of the strands is going to be used as a template to copy and to RNA. And if you look, this RNA has the same sequence as this lower strand, except for that there is a U in place of every T. So if we just read through this, the RNA would read U, G, G, U, 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 G, G, C, U, C, A. And this gets synthesized by RNA polymerase in the nucleus, and as a part of gene transcription. And this DNA template is used, and the RNA polymerase adds complementary base pairs. And the complementary base pair are that A will pair with T, and G's will pair with C's. And so this is being used as a template, and it's unzipped, and so this A is used to, oh, I should say A should pair with T's or U's. And that's the process of transcription. And then that is used, that RNA is used in the cytoplasm by the ribosome for translation. Translation occurs in the cytoplasm. And for each 3 of the nucleotide letters, 1 amino acid is added. So the ribosome uses a triplet letter plan to translate this language from RNA to protein, and each triplet letter is called a codon. And so this shown here, this is the transcription process shown here using the DNA template as a strand to synthesize complementary sequence and make the RNA. And then here's an organization of the RNA as it leaves the nucleus. There's this long region is made up of triplet codons, and the ribosome is going to grab it, and this is a great summary figure of most of Chapter 17. And so the action is here at the ribosome, and as the RNA is slid through the ribosome, there are triplet letters, and you can see there are 3 triplet letters at every given time within the ribosome. And the triplet letter positions are called the A, the P, and the E site. And A stands for the acceptor site, and that's the triple codon in position to add the next amino acid. So here comes the next amino acid, and we'll talk about that in a second. Just go through these definitions. So the first is the acceptor site is where the next amino acid is loaded. So that's where the code is read. There's a genetic code, and this triplet letter calls for the next amino acid. Let's say the next amino acid is alanine, and so alanine is delivered. Then the P site is called the peptidyl site, and that's where the growing protein is bound. And then E stands for the exit site. that's where this factor leaves. And this factor is called tRNA. So we have another RNA to talk about, tRNA or transfer RNA. And here's what happens. Transfer RNAs, there are a variety of different types, and they have a triple letter in their tail that's called the anticodon. And the anticodon will bind to the codon. So that's how the genetic code is read, is that tRNA anticodons bind to the proper pattern of the codons, and the tRNAs bring with them the proper amino acid. And the tRNAs get connected with the proper amino acid by these enzymes just floating around in the cytoplasm, aminoacyl tRNA synthetases. So we have tRNAs and we have amino acids, and this brings them together to produce this activated amino acid that has tRNA bound to it. They can load in the acceptor site. And then once this is loaded in the acceptor site, the mRNA moves over one position, and now this moves over to the P site. And the P site moves over to the E site. The E site is where the tRNA leaves, and it goes back into this pool to have another amino acid added and go through this cycling process again. So this, if we just mark this, here's the position of that tRNA now, and if we move things over 3 letters, it'll be here, and if we move things over 3 letters, it'll be there. So you see the tRNA plays 3 roles. It brings the proper amino acid, it holds on to the growing protein, and then it's released to be recycled. And that's a great summary of the actions in Chapter 17. Here's the genetic code described in Chapter 17. So these are the codons, and all the possible codons have a variety of amino acids they could join to, or there are 3 stop codons. There's a coding region, and a coding region will have these triplet letter pattern until here, where there's a stop codon, and the ribosome will stop coding the protein at that point. The ribosome will let go. And we'll talk a lot more about Chapter 17 in class, because we want to start out with this basic framework and build some on it. We don't need to learn nearly all the stuff in Chapter 17, but we're going to learn some enduring messages from Chapter 17 in protein synthesis.